My name is Peter Tuddenham. I'm with the College of Exploration, and along with the Scottish Association for Marine Science, Anushka Miller, and the University of Illinois, with Professor Gilliam Wood, we put this series together, and this is number 6. We have another one coming up, and we're very pleased today that Catherine Cramer and Steve Uzzo are going to be our hosts from the Woods Hole Institute, and so I'll hand it over to them. Thank you very much.
Great. Thank you, Peter. And good morning, good afternoon, good evening, wherever you may be. Thanks so much for joining us today for this Challenger conversation on art and data visualization featuring Nipam Patel. Nipam is the director of the Marine Biological Laboratory in Woods Hole and is a professor at the University of Chicago. Nipam is also a leading scholar in modern developmental and evolutionary biology with a special focus on the mechanisms and evolution of body patterning and segmentation, regeneration of the germline, and structural coloration. His scientific expertise encompasses the development of novel, genetically tractable model organisms for biological study, which can reveal much about human biology and the application of advanced imaging technologies, which is some of what you're going to hear about today, to probe the fundamental dynamics of living systems. Nipam was also just elected to the U.S. National Academy of Science, and he is also an elected fellow. AAAS, was an HHMI investigator from 1995 to 2010, and has been awarded numerous honors. And since, in the interest of time, I think we will get going, and I'm going to turn it over to you, Nipam.
Thanks, everyone. Thanks everyone for tuning in. And this is a sort of a fun opportunity for me to talk about something that I've always been very excited about, which is, visualizing biology and the intersection that has really with art. And, over the years, I've become familiar with the material that was collected on the Challenger. And I was asked to talk about some of that material relative to what we do with those same sorts of organisms. So if you give me a second, I'm going to set up my screen share here. Okay, so hopefully you all are seeing that just fine. So I was really intrigued by the idea of thinking about data visualization, but really its intersection with art. And I should say that, as a young child, I was really interested in photography, especially nature photography. And so imaging, especially microscopy, was sort of an extension of that excitement. And so hopefully in this talk today, I'll sort of show you an intersection of those two and, how that appeals to me and hopefully appeals to you and the public in general as a way to introduce them to more complicated ideas in science and in research. So I think you all are familiar with, much of the details about the Challenger voyage, which is celebrating its 150th. There's a question. Peter? Oh, okay. Sorry, there was a hand up. And that was a voyage, obviously, that lasted a number of years and collected a huge amount of data, just unbelievable amounts of information from that voyage, and including collecting over 4,700 new marine species. And so that became published in a giant report, and it took quite a number of years before all that information was put together. So it contained, all sorts of information that was really sort of oceanographic, all about ocean currents and salinities and all sorts of amazing information with all the equipment that Challenger had on board. But they also collected an enormous number of specimens, and they preserved them in brine or alcohol. And then when they came back, they actually distributed them to specialists in the field And those specialists spent a long time describing those specimens. And that's part of the reason it took a while to publish everything, because they had to gather all that information. But in describing that, and obviously this isn't something from the Challenger, it's just sort of an old illustration. But of course, this was before there were, cameras to take pictures of everything in the microscope and of organisms and everything. And so scientists really also had to be able to draw, and it's actually an incredible talent. And the biologists at that time did an amazing job drawing the specimens. And I illustrate that here just with an example of, bird illustration. And those of you who have field guides know that many times actually having a field guide that's drawn is actually much better than a field guide that's photographed, because it gives you the opportunity to really bring out the details that you're interested in, especially for identification. And that was especially true of the material that was collected in the Challenger. And so what I'd like to do is go through a couple examples of some of the groups of organisms that were described by specialists from material that was collected by the Challenger expedition, and then show you how we image them nowadays, and how that there's certain, of course, advantages to what we do, even though it's incredible what the drawings were able to show. And then tell you maybe a little bit about the science and some of the things that we're interested in. So I'm going to start with radiolarians. So these are these unicellular organisms that are quite abundant in the ocean. And they are distinctly different than diatoms, but like diatoms, they have a skeleton built out of silica. And they're quite beautiful organisms, so they're incredibly elaborate. And when Haeckel drew these organisms, one of the criticisms he got was that he had made them too geometrically perfect, because because how could single-cell biological things look like this? And one of the things that happened to me in my career is at a certain point I was asked to do an installation at an art exhibit at UC Berkeley. And what I ended up doing was imaging some of these radiolarians and showing that, in fact, they were as or more geometrically perfect than Haeckel had drawn them. So to do that, and you'll see quite a number of images throughout this presentation that are done this way, use a sort of a more modern microscope. So clearly the microscopes that they had back at that time, we would think of them as relatively primitive, but the quality of them was really actually quite good, and they could see quite a lot of detail. And certainly their drawings indicate that they were able to see remarkably fine details and to document them. But our modern microscopes actually allow us to do a lot of other things besides just see the visible light spectrum and take an image. So what I'm going to do now is I'm going to play you a movie, and it's going to show the kind of data we can collect. So right now I'm starting with an image that's just a normal light microscope image. And this would be very much like what Haeckel might have seen when he put the radiolarians into his microscope. The optics here are a little bit different and a little bit more detailed than he might have gotten, but still, you can see how similar it is to Haeckel's drawings. But I'm going to put this movie in motion, and it's going to take a little while to play, but I'll tell you some of what's going on. So we're focusing through it, and you can see that when we have a normal light microscope, one of the difficulties is that we're seeing all the focal planes. But at the same time, you're seeing these green and purple colors. So what we're doing is we're using the microscope to excite molecules in the skeleton and then having them shine back at us. And using that, we can get incredibly good detailed picture that's also very three-dimensional, because we can focus through the object. And then we can use the computer to reconstruct that three-dimensional detail and to create a three-dimensional object. And so you can see now that we can see what Haeckel drew as a 2D, we can really see it in 3 dimensions, and we can rotate it around and we can look at it. And so that's something that's a remarkable advance that we have now. But you can see one of the things that it shows is that, in fact, Haeckel was very accurate. These skeletons of the radiolarians are incredibly geometrically perfect. So here's a couple of the other drawings that Haeckel made from the Challenger specimens. And you can see some of these radiolarians have incredibly elaborate shapes. And some of them look like these bells with these spikes all over them. And here's an example, again, of the more modern imaging on the confocal microscope of one of those other shapes of radiolarians. And again, you can see they almost look like they must be man-made because of the shapes they have. And you can see, again, how accurate Haeckel's drawings were of these things. Here's a couple other, just very quickly, a couple other ones. This one looks like a giant horseshoe. And then finally, there's a few images here at the end. I'll speed it up here a little bit, of some of the other ones. We can also use color to enhance the details that we see. In this case, we're making the different depths of the object different colors, and that allows us to bring out even more detail. Now, people are really interested in studying radiolarians because they're actually quite abundant in the ocean, and they are an important sink of both the silica with which they make their skeletons as well as carbon. And they provide a rich part of the food source of plankton. So I'll move on to some other organisms. One that's near and dear to my heart are amphipods. So these are what we would call beach hoppers if you see them on the beach, but they also live out in the open ocean. And so they're a type of crustacean. And again, different specialists drew some of these amphipods. And so here's some of the drawings, and they have very good illustrations of what this body looks like. You can see they have very odd set of limbs that you can see on these organisms. But one of the things we wanted to do too, in addition to imaging, like I said, on the confocal microscope, which actually is great, but we also wanted to be able to observe animals in more natural habitat. So one of the things we've done at the MBL is build a studio where we can put animals and acclimate them to being in small tanks, but then watch their behaviors and film them. So this is a collaboration with BioQuest, which is an outfit that's in Australia that helps to do all sorts of filming for BBC documentaries and movies and everything. And we've set up a studio at the MBL where we can film some of these. So these are some of these amphipods filmed now. And now, again, the Challenger, the specimens were, brought up and then they were preserved and looked at. But it's great for us to be able to look at them live to see how they actually behave. And one of the things we're interested in is how they use all these different limbs for different functions. So crustaceans have a pair of limbs on every segment, and they use them for feeding and for walking and swimming and jumping, anchoring themselves down. So it's great to be able to have this kind of footage where we can see them live. But then we can also, again, use some of our modern microscopy methods to get very good images. So this is actually taken with what's called a micro CT. It's an X-ray machine that gives us very high resolution. And again, you can see how they have lots of different limbs for lots of different things. And then here's another type of microscope called a light sheet. And again, we can create a very three-dimensional image of the animal and use that in our research. And finally, we can use color to enhance the details. And so here's a particular image I like, and I think it really is on that boundary between art and science. And then we can use modern, again, techniques of integrating genes into the animals to highlight different structures. So in this case, we've been able to insert a green fluorescent protein that lights up the muscle, and you see that here in green, and a red fluorescent protein that's in the nuclei of all the muscles. And again, then we can image the animals. In this case, we can even image them live and see how they're using these muscles inside as they move around, or how these muscles develop during embryonic development. So you can see that in the drawings that people made from the Challenger material, one of the things with crustaceans is they paid a lot of attention to the details of the appendages and the way they're specialized and the way they can be used to identify and distinguish different species. So again, when we image these animals, that's something that we look at quite a bit. And here you're seeing an animal where we've actually lit up the circulation of the animal as well. And then in these images on the right side, you can see both the external part of the limb, which is what you see in those Challenger drawings. But now we can also light up the muscles inside and see the architecture of that muscle. And I have a close colleague, Michaela Savaroff in Lyon, France, who studies how these animals can regenerate their limbs. So you can cut off a leg, and in a very short period, they can regrow the leg, and it looks completely, perfectly normal. And so it's great to be able to see these kinds of structures in detail, understand how that regeneration process works. And then we actually look at live embryos as well. And this is an embryo of an amphipod. And so you're actually looking, the individual dots are individual cells. So we can see how every cell moves around. And in this case, sets up segments of the animal. We've false colored in a couple of cells so you can see their row-like organization as the embryos develop. And then finally, we also have tools to allow us to see how different genes are expressed. This is a set of genes called Hox genes, and in this animal, this is the head of the animal, this is the tail of the animal. And these genes are expressed in this very nice regional pattern along the axis. And we can light up each of the genes in a different color. There's a little movie that shows that. So we've got these 8 different genes, and they're all expressed in different regions. And again, you can see the intersection, I feel like, between science and art. And in fact, it's an art form to really figure out what colors to use sometimes so that you can distinguish all of these different expression patterns. And then we also use CRISPR to make mutants in these things. And so this is an example, again, where we've been able to image it and show this is a normal animal. These red legs are used for jumping. These green legs are used for swimming. And we've knocked out one of those genes, those Hox genes, and we've transformed it so it's lost the legs for jumping and swimming, but it has more legs for walking forward and more legs for anchoring. This is, an example where we can use modern molecular tools to understand details, but the imaging is still vital to being able to see our results and understand what's happening. And we can extend this to lots of different animals. So in the middle, again, is that amphipod crustacean, but on the left is a Drosophila melanogaster, or fruit fly, and on the right is a beetle, and you can see how well conserved the expression of these genes is along the head to tail axis of the animals. And finally, I'll move on to some other groups that were collected by the Challenger expedition. So they collected plenty of sharks, skates, and rays. So here's an example of one of the skates. And there's a scientist at the MBL who works on the skates, various aspects of them. His name is Andrew Gillis, and this is one of the local skates that he works on called the Little Skate. And you may have seen the little purses that skates and sharks are in. So this is a cat shark, and you can see inside. So they're kind of like chicken eggs. They have a big yolk, and the embryo develops on the surface of that yolk. But you can actually take them and open them up and see the embryos very nicely. So this is a young skate embryo growing on that yolk. And here's a little bit of a movie of one of those alive, and you can see its gills are functioning now. It's moving around, and its muscles are going, and it's moving very slowly. But then we can do things like stain for the cartilage. And so all that blue staining you see in this animal is the cartilage. And so its skeleton is made of cartilage. And one of the things Andrew studies is that if you damage this cartilage, it's able to regenerate the cartilage as cartilage and not as bone. So that would be great to understand how that works, because, when we damage our cartilage in our knees, it ends up ossifying, becoming bone, which is not good. But skates can actually regenerate their cartilage. So that's an example of, taking these animals that are in the ocean and actually, doing basic biology, but it has a lot of very interesting potential applications as well. And finally, I'll end with cephalopods. So cephalopods are things such as squid, octopus, and cuttlefish. And so these were also collected and described from the Challenger material. And so here's an example of one of the squids that they looked at. And so one of the animals we work quite intensively on at the MBL is called the bobtail squid. And here's this squid, Euprymna berrii, actually feeding, and you can see it attacking here a shrimp. So again, this is an example of that kind of live movie imaging that we're able to do that's really great for understanding the behaviors of these animals. And here are the embryos. So we're able to watch them developing inside their eggs. So this is a fairly well-developed animal that already looks like a little squid. Here's some younger embryos. So they're on a little ball of yolk, and I think they look kind of like little puppy dogs. And you can see their tentacles here starting to grow, and that's their mantle and their siphon that you're seeing there. And then we can go and image proteins inside of them. So in this case, we're looking at a variety of things that are lighting up their nervous system and their brain and other structures in the animal. So I'm going to play this movie for a little while. It's going to show you various stages of the development of the squid embryos. And you may be able to hear some music. So I think that, again, this is a great example. I entered parts of this movie into a movie contest, actually, that was, at the intersection of science and art. And I think you can see the inherent beauty in many of these things that we're able to see with the modern visualization tools we have, the different microscopes. So again, this is just the light microscope, but you're going to see it transfer over into those fluorescent confocal images where we can generate three-dimensional reconstructions. In this case, we're just seeing the surface of the embryo here. But now we're going to be able to visualize different structures inside of it. In a minute, you're going to see the nervous system of the animal, the developing brain. So we have a couple of researchers here at the MBL, Carrie Alberton and Josh Rosenthal, who actually study how the complex nervous system of cephalopods forms. And they're really interested in using that as a comparison to vertebrate nervous systems. So these are two groups of organisms that have developed really complicated nervous systems. And there's a lot of questions about how similar or dissimilar what has happened in cephalopods for them to have a complex nervous system. And finally, these cephalopods also have this incredible ability to camouflage and to create lots of color. And Roger Hanlon at the MBL studies them. Again, here's a flamboyant cuttlefish, and you can see the amazing colors it's able to produce and the behaviors that it has. It can even modify the texture of its skin. So again, there were some very beautiful drawings of some of these cuttlefish from the Challenger material. And again, lots of detail that we can see, but by our modern methods, we can look inside and see even more. So in this particular example of a cuttlefish, you can see the cuttle bone developing inside. You can see its eyes developing, the tentacles. And then this is the optic lobes in the brain developing inside. You can see how huge it is relative to the size of the rest of the animal. So I just want to thank the people that have provided much of this data. So a lot of the images were taken by students who took the embryology course over the years at the MBL in the summertime. The live videos, again, were shot by BioQuest Studios in a collaboration with the MBL at the studio that we built at the MBL for that purpose. And these are some of the people that have added in I've used some of their images, mostly that they took during the courses here at the MBL. I want to just make sure to thank them. And the Parhayala work is mostly stuff that's done in my own research lab by my postdocs and graduate students. So with that, I'll end and take any questions that you all might have.
Thank you so much, Nipam. Wonderful, wonderful talk. Absolutely beautiful images. And yes, please, anybody with any questions, please put them in the Q&A. I have a kind of an overarching comment. You mentioned quite a few times about the connections between science and art. So, of course, that makes me wonder. And you mentioned something I was thinking also, even down to how do you figure out the colors that you use in some of the imagery? Are there plans, or you've mentioned having the filmmakers there, but are there other plans for producing images of any sort? I mean, this seems like a really fabulous way to get the public even more with the amazing that MBL is doing.
Yeah, one of the great things is that a lot of the microscope companies, for example, will have contests. And I mean, it's becoming more and more popular to have sort of science imagery contests. So, for example, Nikon does a small world contest. And this summer, we'll be, as we do most summers, exhibiting many of those photos and the winners. And it's great fun. And, it's an interesting intersection between the two. And as I said, it's part of what got me hooked on microscopy, was that relationship to photography and art. And again, I think we have to be a little careful. There's visualization that's purely for scientific purposes. And often, in fact, scientists will publish their data as grayscale, which is actually what the data really are. that come off of the microscope because the cameras don't actually see in color. And then we false color them so that when we have multiple things that we want to line up, we can see how they do it. But you can get very much carried away with that too and cross the line into just purely art. But I think even great scientific images that have really high scientific value also can be quite beautiful and is a way to get the public engaged.
Yeah, well, and just to follow on to that, as part of our interest in the Challenger, we did put together, you mentioned an exhibit with your images along with some of the images by Ernst Haeckel. And if anybody's interested in seeing this exhibit, it is still on display at a private residence in Woods Hole, and we do welcome you to come.
And also online at our website.
Yes, and online on the Woods Hole Institute website.
Yeah, and it's a great illustration of the modern imaging techniques next to what are still very quite stunning images that were stunning drawings from the Challenger material.
Yes, so one question I guess I have, as you were going through the slides, there was a picture of two people holding what looked like an Emmy award. And I was just wondering if you can say more about that.
Sure. So the BioQuest folks, they do a lot of imaging for, all sorts of movies and TV shows and things like that. And they shared that for the Puff, which was a movie about the life of a pufferfish. And they were very proud, and it's an amazing feat that they went through to get all that, both a combination of filming out in the wild and filming in the studio. But that had nothing to do with the MBL. That was their own project.
We do have a question for you. How is the concept development of the art pieces beyond just form and color?
Well, I mean, there's the possibility too. I think I'm understanding the question. There's one of the things I've always been excited about is the possibility of printing these things in 3D and having them be stuff that you could actually hold too, and to understand the organisms. And again, it's fantastic to see some of these microscopic things, but then if you could print them much larger, you would, have yet another sensation that you could experience with them, the tactile experiences.
Yeah, that would be fabulous. I've seen 3D printed networks, for example, and just being able to the kind of tactile and turn it around. I think that would be fabulous. We have another question. How long does it take to produce these amazing visualizations? In my experience, new questions can arise when I slowly observe and draw from nature, and I wonder if the more technological production of images also does that.
Yeah, that's a great question. So, you can acquire the images relatively rapidly. It's another amazing example of how the technology is advanced. I remember many years ago doing a microscope confocal image of a cephalopod when I was at the MBL. And I think it was taking like 45 minutes, and it was 2 a.m. And I actually fell asleep and hit my head against the microscope and ruined the image. That same thing would take about 3 minutes now on a modern confocal. So it's amazing how quickly we can do it. But then at the same time, the computers are much faster and they can process it. But that just means that we can get our hands on it faster to really look and stare and understand it. And then also we can do a lot more quantitative analysis. So we can quantitate the amount of fluorescence and understand levels and things like that. So it's great that the technology's made it faster and faster to acquire the data, but then it takes longer and longer to understand it because there's so much of it to look at. And the detail is incredible. And the amount of data, not just the visual data, but down to the quantitation of what you're seeing and the levels and all sorts of things like that, how fast things are moving.
Yeah, and that's a great segue into a question from Gillen Wood, Gillen is one of the researchers at the University of Illinois that really, along with Peter and researchers from SAMS, put this webinar series together. And Gillen says, thank you for the great talk. What do you think has been lost in the decline of freehand sketching skills among bioscientists, given it's no longer a component of undergraduate or graduate training?
Yeah, and I do think that there is a real value to being able to sketch out things, because then you pay attention to details, right? If you're forced to look with enough patience to draw it, then you don't miss a lot of things that if you just, and it's an easy trap to fall in. You just put the specimen there, hit the button, you take a picture, it stores it, you put the next specimen in, and you think you've looked at it because you took a picture of it. It's kind of like photocopying a paper, not without actually reading it. So I do think that, yeah, there is great value in that. And there are still, times when it's good to make a sketch right away and to document those details. So yeah, it is sometimes a shame that we don't put more emphasis on that with students at least when they first start looking at things too, so that they learn to pay attention to a lot of those things.
But it's not currently part of the MBL curriculum.
No, we don't have an illustration course like that. Though as you well know, right, there are very talented people in Woods Hole who still do scientific illustration, and it's fun to have them come to the MBL and do their illustrations of material that's at the MBL, and I find it really valuable. In fact, right now at I believe it's at Falmouth Academy, even there's plankton that's done as needlepoint. And it's fantastic. It's yet another way to, instead of drawing it, but to do it. But it's the same thing. You have to pay all this deep attention to things that you might just overlook if you were just taking a picture.
Great.
So one question I have, I guess, is about thinking about how to take all this amazing knowledge that we can gain from these images, and how can we change the way we educate people about the world? I mean, this is a whole world that you're studying and that Heckle drew. I'd be interested, and maybe others would be interested in how you see that might affect education.
Well, I think one thing, of course, is just, drawing people in with the images, right? So it's, it adds so much more than reading a bunch of text about, describing something, being able to see it in 3 dimensions, or see it live, as opposed to just a fixed object, so, and that's a lot of what we can do now is, I showed just a little bit, but we can do a lot of the confocal imaging live now and see embryos developing, organs forming, and things like that. So I think all of that helps. But, I think the hard part is making that connection, still having the talented people that can take that image and then present it in a way that really appeals to the public. So we make you make a pretty image, but then you still need someone to understand that image, right? So it's always you draw them in and then you explain it. I think a good example is the way NASA, for example, publishes these incredible astronomy pictures, but then they do a great job because they have people that's their job. They write the descriptions to try to draw the public in and educate them, bit by bit about what's so special about what you're seeing.
Yeah, and that kind of reminds me of it might be a little bit too arcane, but these questions about intellectual property, some of which have risen now, a photograph of a portrait of a painting of or a song that originally was an opera that was, there's a lot of this kind of thing from media to media.
Remix.
Right. And yeah. But quite serious intellectual property issues arise. And now that, these beautiful images, and more than images, structural, and particularly if you started getting into 3D printing, for example, it could get even more complicated.
Yeah. I mean, I'm a big fan of making it openly accessible. The difficulty is that if you say you wanted the original data set in order to do 3D printing, the size of the data sets is huge. So there's always this issue of where you're going to store it and how people are going to access it. But it is definitely true. I mean, I feel like, for a long time, I would gather up the images that the students took in the embryology course and store them on hard drives. And it's like, well, what good is this? They're just stored on these hard drives. And so we made a real effort to try to put them up places and have people see them. And then the students get credit for what they've done.
Right. We do have another question. Is there anything to learn in comparing the Challenger images with modern images?
Well, yeah, there's a lot. So, I mean, one of them, of course, was the Challenger in doing this established all sorts of information on diversity and species distribution and things like that, right? And so it's great to have these records so that when modern voyages go out and try to sample, they have a baseline of, well, this is what it was like at this time. And that's really valuable. And then, the Challenger did bring up, like I said at the beginning, 4,700 new species. So it's still fantastic to say, okay, this was drawn and collected from this location. And so if scientists say, well, this particular animal is one that I now have an interest in finding, they know from the Challenger where that was.
Yeah, I guess I have one more question that comes to mind for me is, so it was a fabulous sort of snapshot of where we are now. Where do you see the imaging sort of going in the future, if you can speak to that? And I guess B, sort of what is if you could have any capacity you would want to explore these organisms, what would that be able to do?
Yeah. So there's this, we think of it as this kind of pyramid, that there's always this trade-off between how fast you image, how much light you shine on it, and, how long you can image. And because the light is actually damaging to the animal eventually, right? So, but it's amazing how every bit of that technology keeps improving so that we have better cameras, we have better lasers, and so I think what you see a continual improvement, but you also see some incredible leaps. So you see new methods, modalities of imaging that, for example, are label free. So you don't have to have a fluorescent protein or anything. You just put it in there and you can, using it's called, like, for example, Raman spectroscopy, where then you can see all the lipids. You can make out the outline of everything inside the cell without having to add any label. So then you can take any animal and image it that way. But again, the special equipment is a bit pricey, and the hardware to run it is complicated. But anyway, those are examples where there's lots of new technology emerging all the time. So like now, like light sheet microscopes that actually shine a very, very fine sheet of light through the material, and you can even follow single molecules in living cells now. So I think the sky's the limit. It's almost scary how fast it changes, and any microscope you buy today is, out of date by the time it gets delivered to you, just about.
Got two more questions, Nipam. Hopefully you'll have time to answer them. What do you think of the new trend in which the independent music industry crediting nature as a co-author, which was started by Brian Eno, apparently, I didn't know that, and in so doing, there is a share invested in conservation?
Yeah, no, that's a great point. And it's absolutely true, so you see these amazing images of these organisms, and you have to keep in mind that they live out there in the real world, and that we do want them to be there, for generations to see, but also just on their own right. They deserve to be out there in nature. So I think that's a great point. And I think that's one of the fun things with especially the imaging of marine organisms, because so few people otherwise really get to experience them. But then hopefully they become aware that they're there and they need to be protected.
Right. And one more, is our understanding of marine organisms advancing as fast as our visualization capabilities?
That's a tough one. I mean, it depends on what your units are, I guess, for each of those things. So, um, we are, I think, in a period now where we can explore in ways that we hadn't been able to. For example, by, environmental DNA sequencing and know what organisms are there in the water, even if we don't image them or see them with our eyes at that moment. I don't know. That's a tough one. I think that they're both advancing relatively quickly. I guess, of course, the worry is that there are fewer and fewer people trained classically to look at organisms and identify them. And museums are always under threat of not having funding to maintain their collections. But that's absolutely vital to connecting it to the sort of experiments that we're trying to do now with our fancy tools.
I guess that's all the questions I think we have.
I think you were going to say a few things about unstructured data.
Well, yeah. So one of the things that is an enormous trend going on now, and AI plays a role in what we do with all these images and how we categorize them. I know that there was a big problem that NASA had. I think it was like 10 or 15 years ago, where they actually they started destroying images because they just couldn't store them anymore.
Yeah.
Which is incredibly sad because that's knowledge. So thinking about images as data and thinking about how we extract knowledge from them, which is where my question came earlier from, how do we educate people? Not just about what they tell us, what these images tell us, but also what kind of capacity for understanding the world that imparts on people. I don't know if you have any additional comments about that, but
Yeah, that
these
is a
unstructured
great
data.
challenge. I mean, yeah, even right now, it's hard to store the original data for some of these things, and that is an issue about the reproducibility as well. Yeah.
Well, Nipam, we know that you've got somewhere else you have to go, somewhere
Yeah.
you've got to get to. We really, really do appreciate your time and this fantastic talk, and it will be, has been recorded, and we will send around a link. And we just want to thank you so much. And once again, congratulations on your election to the National Academies.
Well, thank you, and thank you for inviting me to do this presentation and have the opportunity to do something that's actually very fun for me too.
Fun is good.
And an accent, to quote Dr. Seuss.
Yes.
Yeah.
Yes, this has been fabulous. Thank you again. Very kind of you to spare your time, and we'll be interested to see where this all goes.
Yes.
All right, thank you.
And if anybody else wants to stay on for a few minutes, Nipam has to go, but we'll be here for a few more minutes. Thank you, Nipam. So anybody have any other questions they want to post, or are people happy to get 15 minutes back in their day to do other things?
Or to think about imaging and how important it is. And again, this has been recorded and it will be posted online. We'll have a link from our website from the Woods Hole Institute, but also at the Challenger website as well. So.
And again, we do invite you to come see the exhibit it is in a private residence in Woods Hole, but it's still there, and we would love to have you, so please get in touch with us. Maybe we could put an email address in the chat.
Oh, you can go to our website, www.woodsholeinstitute.org, and send us an email.
Great. All right. Thank you all. Have
Thanks.
a good rest of your day.
Peter, any other comments from your side? Okay, I guess we're done. Thank you, everyone.
Thank you all. Take care. Bye now.